Okay, so I want to talk about my most recent project, which involves signaling between cells. So plant cells are connected by plasmodesmata. These are pores that connect the cytoplasm of neighboring cells directly. And through these pores, plant cells exchange nutrients, signals, and this includes proteins and RNAs, including mRNAs, which goes against the sort of central dogma that each cell has DNA produces mRNA produces protein. We now know that this mRNA can move to another cell and produce protein. And recently, it was discovered that some of these mRNAs can actually be transported over very long distances in plants. Some of them can move from the root into the shoot, some from the shoot to the root, and some of them seem to go both ways. So, what is making these mRNAs move and what are they doing? Well, one theory was that they move by chance. So, the more abundant an mRNA, the more likely you'll find it drifting into the other part of a plant. And that seemed like it made a lot of sense. It was then, however, upended completely by finding out that some mRNAs are only mobile some of the time. So the gene CK1 produces an mRNA in two versions. One of them is the normal version transcribed in the normal way, and one of them reads through and includes a nearby tRNA gene, which means that one of the versions of mRNA has a tRNA at the end. Only the one with the tRNA is long distance mobile, and it can move right from the roots to all the way up to the flowers in Arabidopsis. So what this means is that mRNA mobility is not just random, not due to chance. There are motifs and mechanisms that can be added to cause mRNA to be transported, meaning that this is a deliberate signaling mechanism. So to test this out, what we did was we took these tRNA motifs and fused them to CRISPR-Cas9, to the Cas9 and to the guide RNAs. We wanted to see if we can make a long-distance mobile gene editing system. So the idea being you graft two plants together, the Cas9 moves from the root into the shoot, edits the flowers, and the seeds produced from there in the next generation are edited, and it doesn't need a transgene in the shoot at all. And we found that this does, in fact, work. It turns out that having a massive Cas9 mRNA with a tiny tRNA on the end is sufficient to make that mRNA mobile all the way to the other end of the plant, be translated into a functional protein, and cause gene edits. So this has therefore unlocked a whole new dimension of cell-to-cell signaling of plants over really long ranges and raised some really interesting questions. How does this mechanism work? What other RNA motifs are there? What is the function of this long-distance mRNA signaling? How does it control plant growth and development? And with a little luck, I will be able to answer some of these questions during my career and hopefully in the next question session. Thank you.